I'd like to begin with this title. Oxidative stress refers to a condition in which reactive oxygen species oxidize cellular membranes and proteins, leading to a decline in normal biological functions. It is known to be contributing to cardiovascular disease, cancer, and aging-related disease. Therefore, antioxidants are attracted attention because they scavenge activity for loss. Loss are divided into logical and non-logical species in vivo. Logical loss like superoxide anion and hydroxyl logical are mostly made in the mitochondria. Non-logical loss, such as Singlet oxygen, are made when the body is exposed to UV light. Since these losses have different properties, antioxidants need to work against both types to be truly effective. So we aim to clarify the loss type specific reactive of antioxidants and explore those with the potential to scavenging a wide range of loss in vivo. We use 5 different antioxidants. They can be classified into two main groups. First group is carotenoid, which are red to yellow components found in shrimp and vegetables. Specifically, we use hydrophobic carotenoid, including astaxanthin and beta-carotene, and also a water-soluble one called Crocin. Second group is vitamin E, which are existing biological system. We also use hydrophobic alpha-tocopherol and hydrophilic torox. So, first is organic logical quantum assay using electron spin resonance to measure logical scavenging activity of antioxidant. ESR measures the types and concentration of logicals in the sample. It is known that the intensity reflects the amount of logicals, so we're focusing the amount of logicals and the antioxidant relationship. Second is Singlet oxygen absorption capacity. The absorption spectra of the ligands used are shown below each chemical structure. N-peroxide generates singlet oxygen at 35 degrees. The generated singlet oxygen is subject to a competitive reaction between the probe compound 1,3-diphenyl isobenzofuran and antioxidant. By observing the decomposition rate of DPBF, we evaluate the singlet oxygen scavenging activity of the antioxidant. Here is the result of the logical scavenging activity. In this study, we used DPPH logical and galvanoxyl logical as the logical species. Galvanoxyl logical is the more hydrophobic than DPPH. In this case, I will explain the result for alpha-tocopherol. This graph shows the relationship between intensity and the concentration of alpha-tocopherol. As the concentration of alpha-tocopherol increased, the intensity decreased. This indicates that alpha-tocopherol is scavenging the logicals. To evaluate the scavenging activity, we calculate the EC50 value. EC50 represents the concentration of antioxidant required to reduce the initial logical amount by half. EC50 result. Please take a look at the graph below, which shows the logarithmic plot of the inverse EC50 values. Vitamin E shows high scavenging activity. However, hydrophobic carotenoids such as astaxanthin and beta-carotene show no activity. Interestingly, Crocin was the only carotenoid that showed scavenging activity. This result suggests that logical scavenging activity depends not on logical polarity, but rather on the type or group of antioxidant. As a result for singlet oxygen. In graph A, the time-dependent change in the absorption spectrum contains only end-peroxide and DPBF. As singlet oxygen is generated over time, DPBF is decomposed and decreased in absorbance at 430 nanometer is observed. In contrast, graph B shows the spectrum when beta-carotene is added. The decomposition of DPBF is suppressed. However, since the absorption bands of beta-carotene and DPBF overlap, it is not possible to track the amount of DPBF decomposition. To solve this issue, we applied singular value decomposition to isolate the spectrum change of DPBF, monitor its decomposition. In this graph, the vertical axis represents the amount of DPBF decomposition, and the horizontal axis shows time. The decomposed amount of DPBF was reduced depending on the beta-carotene. So the reaction rate constant K for the reaction between DPBF and singlet oxygen was determined using a fitting equation. Since we were able to calculate K, we used the analysis equation to determine the reaction rate constant Kq between singlet oxygen and the antioxidant. In this equation, the vertical axis presents the reactivity PBF decomposition rate in the presence and absence of antioxidants, while the horizontal axis shows the antioxidant concentration. The slope of this plot reflects the singlet oxygen quenching activity. In this study, we found that when plotting against the square root of the beta-carotene concentration, the dates show high linearity. This suggests a half-order reaction in which one molecule of beta-carotene quenches two molecules of singlet oxygen. Based on this result, we applied the same type of plot to other antioxidants as well and compared KQ. In this graph, the vertical axis represents the logarithm of the KQ value. For singlet oxygen, vitamin E like Trolox and alpha-tocopherol showed very little activity. In contrast, astaxanthin and beta-carotene show strong scavenging activity, and Crocin showed moderate activity. Just like with logical scavenging activity, this result suggests that the singlet oxygen scavenging activity depends on the type of antioxidant too. In conclusion, the antioxidant used in this study can be broadly classified into three groups. The first group is radical quencher type. This includes vitamin E, such as Trolox and alpha-tocopherol. The second group is the singlet oxygen quencher type. This group includes hydrophobic carotenoid, such as astaxanthin and beta-carotene. The third group is Crocin, which shows high activity against both radicals and singlet oxygen. Crocin's unique dual reactivity still requires more investigation, but our findings suggest that crocin can scavenging broadly loss in vivo.